Be some folks deciding they're going to just stay.
Just how you got all the examples? You must have all the examples. Well, then get to work. I'll call on you first.
All right, are we ready?
Josiah's going to share his first example. I'm sure you can. Collagen, that's the one from 2 days ago, right? That was the example of the protein that fills up every space, and it also makes up glue. So, which is also why glue used to be made from horses, the same way Jello is made from cows. So, collagen. Go ahead, Josiah. Catabolism, right? And so that is like a reaction that breaks things down, and that's a good one. But I am going to combine it with something else right here. Catalyst, right? Because that means to break things down and dissolve things, and it is one way of describing some things that enzymes do. So we can put that one for both of them, a catalyst. Just real quick before I forget, I want to keep up. That. I'm gonna make a mess in a little bit too. This one's gonna go. All right, good. Somebody else. Mitosis right here.
Yeah.
That's mitosis, and it doesn't have anything to do with muscles. It does have to do with cell dividing, and we'll get to that here in another 2 minutes, I think. So that one's not going to work. The reason we have that, I gave you guys 3 examples of proteins, and I could have given you like hundreds, right? But there's one other one that you might hear sometime. It is myosin is the muscle and protein. Is the protein and muscle that we eat. It's the red fibers in a steak. It's what allows muscles to contract. That's one of the more abundant proteins in the human body, and so I thought it would be a good one for us to have. We talk about that in our heads. Others? Decompose, to break down, get broken down, right? We're going to use it in a very similar way, but this is specific to enzymes, and it is called to denature, to undo the normal thing that an enzyme is supposed to do. Keratin is another one of the examples from last class. I'll close this down in case they have any questions. I can't hear them because the volume is off. There we go. Oh, and it's also what horns are made of, in like a cow horn or something like that. Same thing, hair and cow horn. Submarine to be underwater. We are going to combine these two right here, and it's not a great direct translation. I guess it's changed a lot over time, but the word is substrate, and instead of meaning below the layer, it means before the arrow. And so like it's not a good direct translation. But we're going to use that today as we talk about things before the arrow in an enzyme reaction. You're probably going to have to use Google for this one unless you know one. An enzyme ending in ASE.
Oh.
You can have Google.
I have it right here, but I don't know how to do it.
You can try. That's all I've ever asked. Try that. Amylase. That's a great one. So amylase breaks down amylose. What did you have?
Peroxidase.
Peroxidase is one that we're going to be dealing with today. I'm going to put the most simple one, catalase, because it turns out enzymes are named after the thing they break down. Like, what do you think you would call the enzyme that breaks down lactose? Yeah, you just change the O to an A. Amylase, what Stephanie found, breaks down amylose, which is another word for starch. Catalase was one of the first ones discovered, And because of that, they just named it, oh, it's this thing that breaks stuff down. We could also call it peroxidase as a more technical term, but the word catalase is a really common one to use as we talk about enzymes, okay? So these will be on the test. List 17, 16, and 15 are the ones from this semester. And I know today is only the second day of the unit, but for you guys, I believe that would put us at next Friday for that test. That also we have a vocab portion. I don't know if I have any other vocab to do, and it looks like I don't. So we won't be having any vocab until we get to DNA and the cell cycle next unit. So, all right, that means we need to. Oh, we're going to take out our notes, and you guys can go too. And the demo is kind of part of the notes. So let's go ahead and take out the notes here. There, I got the wrong writing. The wrong thing. Wasn't the light on? Are you looking off here? Direct instruction time, but we're going to see stuff. Here's my joke. Steve gets it.
Oh, because H2O2 is like it's toxic.
It's toxic, right? Not H2O also, but H2O2. And you guys probably have it at home. And you have it in little brown jars like this. Hydrogen peroxide, okay? So this is hydrogen peroxide, and the chemical formula for hydrogen peroxide is H2O2 instead of H2O, okay? This is 3% hydrogen peroxide, and you probably have it at home for one of two reasons. Why would I use it?
Like disinfectant.
Yeah, cuts and scrapes and stuff like that. And when you pour it on your skin, it bubbles, which is what we're talking about today. It's also a really cheap mouthwash because it kills bacteria pretty well. This is like $1 for this right here. It doesn't taste like it tastes like bubbles. But you probably have it at home, and it always comes in a brown bottle, which we will talk about.
Wait, you can use that for mouthwash?
Yeah.
Wait, if you die, why?
Well, because this is 3% hydrogen peroxide, which is fine. And a lot of them are even sold with mint flavor. Like if you go next to in the store, you'll see this one. You'll see a blue one that's got like mint. But anything that gets higher than like 10 to 30%, like above 30%, you shake it and it explodes. This is 3%, and it's really pretty safe. Right? I'm not saying somebody should go drink this, but one of these suggested uses is, let's see, first aid to help prevent the risk of infection in minor cuts. Aids in the removal of phlegm, mucus, or other secretions associated with occasional sore mouth. And I guess so yeah, it is toxic, but it's one of those things that the amount is all that really matters, right? A whole bunch of this, and that's a bad thing, but the volume that you use. All right, so clearly we're dealing with that. We're also dealing with this. What's that? Yeast, right? So I've got yeast, and I have so this is a living thing. This is why I needed chicken blood, because chicken blood still has the active enzymes in it. But so do yeast cells, and I woke these guys up like an hour and a half ago before the last class. And so they are now alive and growing and dividing. They're packed with a little bit of sugar in there. And their enzymes have, when you put water on them, been active. They go dormant when you dry them, which is why this stuff is good for like a year and a half or something like that. It'll last for a long time when you put water on it and they wake back up. And so I have a toxic thing that all cells produce that's bad for those cells. And then I have living cells that we're going to put into contact with that toxic thing, okay? So, and it's a visible reaction, so I'm just going to do it right here. About that much. I'll hold it up for you guys on the computer too in a second, so. Cool fingers. There we go. All right, so we take the yeast, a few billion cells, and we add them to the hydrogen peroxide. Let's see what happens. It's nothing but crazy, right? But it is certainly a visible reaction. We can see bubbles being produced and rising up. bubbles. Bubbles. There we go. All right. So we can see the bubbles. Okay, but then we can start asking a whole bunch of questions, right? Like, once the bubbles stop, at least, if I can speed up a bit this. We're not quite done yet. Maybe another one for stirring purposes. Have we stopped? All right, so this doesn't have anything. Okay, so they're, for the most part, they are stopped. Speeding it up a whole bunch. Okay, so the question I want to ask is, what's going to happen if I put more yeast in? You have a guess. It'll bubble up more. It's a perfectly valid guess. I'm afraid it's wrong. Okay? That doesn't make much sense, right? You mix the two together and then it bubbles up. Well, what if I put more of this in? Then it should. And that'll tell us something, and it takes a second to get going. And there it goes, right? More bubbles. There we go. I can show you the speed up. So that tells us, what is the source of the gas that makes the bubbles? Does the gas come from this, or does the gas come from this? Yeah, this is what makes it show up, right? But it comes from this. And so, like I can add a whole gallon of this, and if there's none of this, it doesn't make any difference, right? But that also means if the yeast is in there making these bubbles, right? The bubbles seem to be just about done. They're close to being done. I'm not going to add any more yeast. Can I add more of this, or did that thing, whatever's in here, did it get used up? Try again. Bubbles. So it didn't get used up. Is it used up now? Find out. More bubbles. It seems like no matter how often I do this, whatever it is that's making the bubbles does not get used up. It sticks around and it can do the reaction again and again and again. And I could do this 100 times and it would keep going and it would keep going and it would keep going, right? And so it's not getting used up, whatever that thing, whatever the enzyme is, the enzyme called catalyst that breaks this stuff down. So, all right, but I boiled this one here a while back, right? And so I put it in there when y'all were doing the vocab or maybe just, like, I don't know, sometime around there. And so now this yeast has been boiled for, I don't know, 10 minutes or so. So I can put these two, one right here. I'm gonna move it back just a little bit because sometimes we get a mess. I don't know why I have it there. Okay, so, and then I'm gonna get equal amounts of peroxide. What is by the way, this is the one that was boiled, right? What's that gonna do to the ability of whatever it is in here to make bubbles? You guys got decrease it, kill it. Well, I mean, those yeast cells are alive. The enzymes are not alive, but they could certainly have been killed. So then, let's see. It seems like when you heat enzymes up a lot, they no longer function all that well, do they? Something happened when I boiled it for just about 10 minutes, and it doesn't work anymore. And it's really easy to understand what's going on. Enzymes are protein, so is a steak. Okay, when you heat up a steak on a grill, does it look the same after it's done? Does it change color? Has it changed shape?
Yeah.
How did it change shape?
It got sweetened.
It got smaller. Didn't I say yesterday that proteins and their function is entirely based on their shape, right? They only do what they do because they have certain shapes. And if you change that shape, they don't do what they do anymore, which is what I did. I changed the shape of these by heating them up. It takes all the proteins and makes them expand and then contract down to the small. So this one still works just fine. I just made a few messes. I'm going to clean that up in a little bit. So that's a little introduction to enzymes, right? It shows you a chemical reaction happening. It shows you enzymes doing that. It shows you a little bit about them. It shows you that I can still keep adding. And it goes a little bit slower, but there's bubbles. So they're not getting used up as they're doing this. It shows you that the bubbles are coming from this, but this is what's making them show up. So there's a lot you can kind of learn from just this little introduction demo to enzymes that really helps us understand how enzymes works. Okay. And we'll do a lab in 2 days when y'all are back here that is like a modeling lab of breaking toothpicks where you can learn some more stuff about the speed of a reaction. But what I want to start off with is this, the definition of enzyme. And then I want to write or draw this reaction in 3 separate ways. I want to do like a picture diagram. I want to do all reactions, and then I want to do this reaction. Okay, so let's move on to, I'll clean that up while I'm doing the assignment. So let's move on to the diagram. Okay, if I was to diagram one of these reactions taking place, we could do something like this. Something like that. And something like that. What do you notice about the shapes of the red and blue things?
They're basically.
They're like, I wouldn't say they're the same, right? But they match up. They're like a puzzle piece, right? And it's often how it's described. It's also called the lock and key model of enzyme reactions. And so the point is that they go together like pieces of a puzzle. And so shape, it is a 3D shape that matters when you're talking about enzyme reactions. Now, if we wanted to talk about like a specific reaction, or not, if we wanted to talk about all reactions, there are names for the thing that shows up before the reaction. That is called the substrate. The substrate means the exact same thing as reactant. It just means in an enzyme reaction, it is called a substrate. I don't know why they picked something different. So, what goes to the right of the arrow in a chemical reaction? This one hasn't changed. What we call the thing after the arrow? Product. So that means we have up here the substrate and the product. Where are we going to put the word enzyme? In the middle, right? Because it's not used up and it's not produced. So we put that right there. So anytime you're talking about these, there's a substrate, there's an enzyme, and there's a product, and there's an arrow connecting those two things. What about this reaction? Where we're going to put hydrogen peroxide? Is that substrate? Yes, good, because that's the thing we start with, right? That was H2O2. Where am I going to put catalase?
At the reaction.
No, because that's the subject and the reaction at the same time.
Oh, I mean, no, product.
Nope. On the line, because it's the enzyme, remember? It's an AOC. So catalase or peroxidase, as we came up with as well, which they mean the same thing. Catalase goes right there. Well, then think about, okay, this image, this diagram is showing it being broken apart into 2 pieces. Well, what are 2 non-toxic things that you can make from H2O2? One of them being a gas that would make bubbles. If this is bad for cells, what 2 things can we make from it that would not be bad for cells, one of which would make bubbles? And? Yep. H2O plus. The problem is O2, it's always diatomic. It's always like paired up with another thing, and so it has to be O2. This isn't balanced. I think I'll balance it. Right now I have 2 hydrogens and 2 oxygens on the left, and then 2 hydrogens and 3 oxygens on the right. Next year you will learn about the process behind balancing equations. Remember, you put coefficients. You put coefficients in front right there.
Wait, then you should put like ones.
Ones are already there, right? Like in the same way, like if you have something like 2 plus 3 right there, that implies there's a 1 right there because it doesn't change anything, right? So we need to put something other than 1 in front of some of these things.
So you said it has 2 hydrogens?
On the left, there's 2 hydrogens, and on the right, there's 2 hydrogens.
So you put 2 in front of both of them?
In front of both of these. This and this. Now, how many hydrogens are on the left? 4. And how many hydrogens on the right? Yeah. How many oxygens on the left?
2.
No, distribute the 2.
Oh, okay. 4.
4. And then how many oxygens on the right?
2.
Nope.
4.
4. Because you distribute this 2 to right here, and then you have 2 more right there. So now it's balanced. You were right. You got it balanced. It's a 2 in front of water and a 2 in front of a 2 in front of hydrogen peroxide and a 2 in front of water. Okay. So that is now a balanced equation. This is what we just saw. This is how we do all of them, and this is the visual diagram, a common thing to see in like a textbook or picture or something like that. That's enzymes. There's a lot of other stuff that you can cover with enzymes. What I really like to cover on the first day, before we get to like in 2 days, we're going to cover factors affecting enzyme reactions, enzyme concentration, substrate concentration, inhibitors, denaturation, things like that. I think it's really important to think about energy and why sugar doesn't constantly explode. You've got sugar at home and it just explodes into fire. But kind of it should, right? It stores sugar, it releases energy, and what's keeping it from doing that? So I want to draw 4 graphs right here, okay? 1, 2, and then I'm going to leave the bottom blank for a moment, okay? Both of these graphs, all 4 of them technically, are going to deal with CO2 plus H2O producing C6H12O6, and then going the other direction. And I picked this reaction. I could have picked tons of different reactions, but I picked this reaction because we just covered it 2 years ago. What is the arrow on top pointing to the right represent?
ATP.
Nope. What reaction?
H2O.
That's not a reaction. You're telling me molecules. I want to know what the reaction that is that makes sugar from carbon dioxide and water.
Wait, CO2 and H2O.
Say the name of a reaction. Making sugar from carbon dioxide and water. Let's do it.
Oh, photosynthesis?
There we go. That's a reaction. Well, then what was this reaction going from glucose back to CO2 and water?
Dehydration.
Nope. After photosynthesis, we learned the second half of the carbon cycle. The energy is released from sugars and used to make ATP. It involves usually.
Cell respiration.
There we go, cell respiration. All right, we got there. So this is going to be photosynthesis, and this is going to be cell respiration. Each of them are going to have time of the reaction right here, and energy right here. Okay, so I want you to think. If you had, on one hand, 10 grams of carbon dioxide and water, and on the other hand, you had 10 grams of sugar, they would not have the same amount of energy in them because of the complexities of bonds and how those bonds are formed. Which one has more energy, 10 grams of sugar or 10 grams of carbon dioxide and water?
Sugar.
Yeah, because the whole point of making sugar is to store energy. And so it turns out with photosynthesis, you get an uphill reaction. And this is C6H12O6. And a ball will not roll itself uphill. That's what sunlight does. It pushes the ball uphill. So we can make a high energy molecule, right? We've kind of done this before, but we're looking at it in a different way. Well, over here for cell respiration, sugar to CO2 plus H2O, won't that ball roll downhill?
Yeah.
And that's combustion, right? Turning sugar or carbohydrates into things like CO2 and water. So if we have a pile of sugar on a table, why doesn't it, if it's constantly, trying to roll down that hill and release the energy, what stops it from doing that?
Constant supply.
Well, but sugar has the energy stored in it, right? We're going to figure it out. Before we do that, let's try to give names to these. This one right here. Are we going to call that exothermic or endothermic? Think about what those terms mean. Therm means energy or heat or, like fire, that kind of stuff. One of them is going to release energy out to the universe. And one of them, the universe has to put energy into it. Good, because it releases energy. Burning sugar releases energy. This is, and then this one is There's a really easy exothermic reaction that I can demonstrate. I'm not going to finish the reaction, but think about this. A ruler that is intact, and then a ruler that is broken into two, okay? So I'm going to put a butt right here. Oh, it's scratching. Time, energy, downhill reaction. Okay, well, that's about it. 2 dots that go down. Okay, so that's what this would represent, breaking a ruler. But as I start to break the ruler and I start to bend it, what happens to the amount of energy stored in the ruler as it goes from not being bent to bent and right before it breaks?
It goes to the same.
Well, but what happens to the amount of energy stored in the ruler as I do this?
A lot more.
There's more because it's under tension, right? Like I'm putting energy into it. Eventually, it's going to break and it's going to go downhill to the lower energy version, right? Because I'll have 2 pieces of ruler. But temporarily, there's a lot of energy stored in here because I'm bending it under tension. So the more accurate picture is that. There's a bump. That bump exists over here, but it's not really as necessary because like it still is. And it's an uphill reaction with a bump. I'm curious more about this downhill reaction, and that's why if you have a pile of sugar, it will not explode because you have to get it up to the top right here, and then it can roll down. And if you want to burn sugar, what do you have to do in order to get it to burn? Heat it. Put a match to it, like light it on fire. And then that match, that heat will put that energy and that sugar up over the hill, and then it'll fall down the hill, right? That is called, that amount of energy is called activation energy. This reaction that I showed right here, it has an activation energy too. And that explains why at home, your hydrogen peroxide is guaranteed to be stored. Why is this always in a brown bottle?
Oh,
So like,
Yeah, because this is a really small bump. The activation energy for the breakdown of hydrogen peroxide into water and oxygen gas is a super small bump. Doing this adds enough energy for me to break down a little bit of it, right? And sunlight adds plenty of energy to break down all of it. So the only way to keep this from turning into water and oxygen gas and pressurizing the bottle is to put it in a brown bottle. So you have to always keep this in a brown bottle. The other thing you can do is mix it with the yeast, right? So yeah, go ahead.
all right. So if you put it in like a water bottle and you put it on the sun, it will explode.
I don't think it would pressurize that much because that's only 3%. And so the amount of gas that would go into there would not be very much. However, it would pressurize the bottle, and it might pop the bottom out kind of thing or something like that. I bet it would. But no, I don't think it would turn into like an explosive like bomb kind of thing. But it would definitely pressurize it. Okay, so think about the enzyme, right? The enzyme, if we change this all of a sudden to H2O2, and then H2O plus O2 right there, did the enzyme change what we started with? No, right? It's still hydrogen peroxide. Did the enzyme change what we finished with? It's water and oxygen. How can the shape of this line change to make it easier to get from here to here? If the bump is lower, that's exactly what enzymes do. Enzymes lower activation energy. And then make it easier to get over the hill and turn the substrate into the product or the reactant into the product. But they don't change what the reactants and products are. They just make it a lot easier. Okay? That's what enzymes do. And they do it because they have a shape that is like halfway in between the substrates and the product. Okay? Now there's a lot more to cover with enzymes. Now when you think about like the test coming up next week on enzymes and proteins, this is complicated stuff. One of these graphs is guaranteed going to be on the test, but the vast majority of my questions will come from the general ideas on this, with like what is a reaction, what do these things do, predict what's going to happen if we do this and that kind of stuff. But my AP students do a lot of math with this kind of stuff, and so I did want to make sure that we saw this because this is like moving it. So my AP might higher level biology class. All right, go ahead and close that and open up close your notes and open up the homework, and we'll make sure you know what to do. I got a ton of time. Good, because I can go over grades and attendance with everyone. So I always like to do one of the reading assignments at the beginning before we get into any sort of virtual labs or mini labs or something like that. And that's exactly what we have this time. This is an assignment from the reading, McGraw-Hill reading. It is chapter 6, section 6.2, pages 156 through 160. That is called chemical reactions. The majority of that reading is not about enzymes. It's about the general idea of reactants and products and balancing and chemical reactions. The last part of it does get into enzymes, and so you'll have to fill in 1, 2, 3, 4, 5 different things about enzymes. It's not very hard. It shouldn't take you very long, but at least it gives us you seeing the textbook and seeing what we're learning about with enzymes. So this is a grade that is due for you guys on Friday. When we'll come in and we'll do the toothpick-based lab. Okay? Questions for me? If you didn't finish the reading assignment, you got to do that one first. Okay? Or not the reading, the writing assignment. So do the writing assignment first. I'll take a look right now and see how it's going. On to all right, Ella and Jolynn, we're just doing the homework reading assignment now, okay?
Okay.
I'll see you later.